Right now, you are burning energy at the rate of a 100-watt light bulb. Not because you are moving, not because you are thinking, but because if your temperature drops by just 2 degrees, your heart could stop. And if it rises by 3, your proteins begin to fall apart. Inside you, there is a silent war against physics. Every second, billions of mitochondria leak energy on purpose. Blood vessels open and close like hidden valves. Fat cells ignite. Muscles tremble microscopically. You are not passively warm. You are being actively defended. And in the last few years, scientists discovered something unsettling. Your immune system talks to your thermostat. Your fat cells can change identity. And your core temperature follows a circadian rhythm, even when you don't notice. We usually say 37 degrees Celsius is normal, but it's actually a pretty cool temperature. We call 37 degrees Celsius normal. Modern continuous core temperature studies show subtle oscillations across 24 hours, up to 1 degrees Celsius difference between early morning and evening. This rhythm is driven by the suprachiasmatic nucleus, your circadian clock, which synchronizes thermoregulation with metabolism, cortisol release, and mitochondrial efficiency. We usually say 37 degrees Celsius is normal, but it's actually a pretty cool temperature. Your temperature rises before you wake up, not because you moved, because your clock predicted you would. They sense temperature directly through transient receptor potential channels, molecular thermometers embedded in lipid membranes. Deep inside the preoptic area of the hypothalamus, specialized neurons do something extraordinary. When temperature shifts, membrane fluidity changes. Ion permeability shifts. Firing rates adjust. But here is what recent research uncovered. Microglia, the brain's immune cells, modulate these thermosensitive neurons during systemic inflammation. In 2023 to 2024 studies, neuroimmune crosstalk showed that cytokines do not merely trigger fever indirectly. They reprogram hypothalamic circuits through local inflammatory signaling, altering neuronal excitability. For decades, we believed brown adipose tissue disappeared after infancy. Fever is not just a chemical signal. It does not. Advanced PET imaging and single-cell RNA sequencing revealed metabolically active brown and beige adipocytes in adult humans, especially around the supraclavicular region. But the bigger discovery? White fat can transform. Under cold exposure, specific progenitor cells within white adipose depots differentiate into beige adipocytes. These cells upregulate UC1 and acquire thermogenic capacity. Fat changes identity. At the molecular level, sympathetic stimulation through beta-3 adrenergic receptors increases cyclic AMP, activates pKa, and triggers transcriptional programs via PGC-1-alpha. Mitochondria proliferate. Proton gradients become deliberately inefficient. Energy becomes heat. You sacrifice ATP efficiency for thermal survival. Evolution selected controlled inefficiency. Your skin is not passive insulation. Recent endothelial studies show that cutaneous blood vessels express thermosensitive ion channels themselves. They can locally adjust vasodilation independent of central commands. Nitric oxide synthesis increases. Smooth muscle relaxes. Heat radiates outward. Sweat glands, innervated by sympathetic cholinergic fibers, create evaporative cooling that makes humans the most effective endurance thermoregulators among mammals. This is why Homo sapiens could outlast prey in the savannah. Persistence hunting required thermal mastery. Civilization began with temperature control. During infection, macrophages release IL-1 beta and IL-6. These cytokines induce prostaglandin, E2 production in the hypothalamus. The thermostat resets upward, but newer findings show that peripheral immune cells also alter brown fat activity directly, enhancing thermogenesis during early infection. The immune system and metabolic system cooperate. Raising temperature accelerates immune cell trafficking, improves T-cell kinetics, and may reduce pathogen replication efficiency. Above 41 degrees Celsius, protein tertiary structures destabilize. Hydrogen bonds weaken. Cytoskeletal integrity falters. Heat shock proteins, HSP70 and HSP90, bind exposed hydrophobic regions, attempting refolding. But in heat stroke, gut epithelial tight junctions fail. Endotoxin leaks into circulation. Systemic inflammation spirals. It is not heat alone that kills. It is immune collapse triggered by thermal disruption. Fever is strategic biophysics. Below 35 degrees Celsius, enzyme kinetics slow dramatically. Ion channel conductance decreases. Cardiac pacemaker activity destabilizes. Calcium handling in cardiomyocytes becomes erratic. Ventricular fibrillation risk rises. Coagulation cascades lose enzymatic precision. Temperature is the speed limit of biochemistry. Reptiles borrow warmth from the sun. You manufacture it internally at enormous metabolic cost. Endothermia allowed stable enzyme kinetics. Stable kinetics enabled sustained aerobic metabolism. Sustained metabolism allowed brain expansion. Your intelligence depends on thermal constancy. Your thoughts exist at 37 degrees Celsius. You are not warm by coincidence. You are warm because billions of molecular decisions are made every second. Ion channels open. Fat cells ignite. Blood vessels negotiate. Immune cells adjust the thermostat. Your circadian clock predicts tomorrow's heat. You are not surviving temperature. You are continuously engineering it. 37 degrees Celsius is not a number. It is a biological achievement.